Hello everyone, it's Tutor Mifzal here, and today we are going to cover the structure of the plant cell and its organelles. But before we do that, do follow me on my Facebook and Instagram at Tutor Mifzal, and don't forget to like, share and subscribe my YouTube channel. So now let's begin. So the plant cell is similar to the animal cell except for a few additions of organelles which the animal cell does not possess. Similarly, we are going to draw the plasma membrane. First thing that we are going to draw is that we are going to draw the plasma membrane, and the main function of the plasma membrane is to regulate the movement of substance in and out of the cells. So we'll just label it plasma membrane, and we will jot down the function of the plasma membrane, which is to the next structure that we are going to draw is the cell wall. So let's label it as cell wall. The cell wall is made up of a tough substance known as cellulose, and it provides mechanical support and protection to plant cells. It also maintains the shape of the cells, and lastly it prevents the cells from bursting. So we'll just jot down all the three functions of the cell wall. Alright, the first one is to provide mechanical support. The second function is to maintain the shape of the cell. And last but not least, the main function of the cell wall is to prevent bursting. Bursting is caused due to the high turgor pressure due to osmotic pressure. Alright, the next structure that we are going to draw is the nucleus. Similarly, like the animal cell, the nucleus consists of the nuclear membrane, and inside the nuclear membrane we have the chromatins, and in the middle of the nuclear membrane we have this dense structure known as the nucleolus. So the main function of the nucleus is to control all activities of the cells, and it contains the genetic information of the cells. And next structure that we are going to draw is that we are going to draw the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So this is the rough endoplasmic reticulum. To differentiate between the rough endoplasmic reticulum and the smooth endoplasmic reticulum is that there are ribosomes attached to the rough endoplasmic reticulum. So what we are going to do is that we are going to draw the ribosomes first. So we'll use this dark blue color. So this is the ribosomes that attaches itself to the rough endoplasmic reticulum. In some cases, ribosomes are scattered everywhere inside the cells, and the ones that attaches itself to the rough endoplasmic reticulum is mainly used to produce extracellular enzymes. So this is the rough endoplasmic reticulum, and we have the smooth endoplasmic reticulum. We label it as the rough endoplasmic reticulum. And the main function of the rough endoplasmic reticulum is to transport protein synthesized by the ribosomes. Next thing that we are going to do is that we are going to label the ribosomes and its function. So this is the ribosomes. The main function of ribosomes is for protein synthesis. So what happens is that the nucleus contains the genetic information of the cells, and inside the nucleolus is where RNA is produced. So what RNA would do is that RNA would decode or decipher the genetic information from the chromatins, and after that it would attach itself to the ribosomes near the rough endoplasmic reticulum, where at the rough endoplasmic reticulum the ribosomes would translate and make the instructed proteins based on the instructions from the RNA. When the protein is synthesized, it will travel to the rough endoplasmic reticulum, to the furthest part of the rough endoplasmic reticulum, so where it will be encapsulated in a transport vesicles. This is a transport vesicle, and when the transport vesicle travels, it would travel to the Golgi apparatus. So this is the Golgi apparatus. The main function of the Golgi apparatus modify, process, and package proteins from the transport vesicle from the ribosomes over here. Alright, so what happens next is the modified protein would be encapsulated in a secretory vesicle, and it would travel to the plasma membrane, where it is going to be secreted out of the cells in a process known as exocytosis. The next structure that we are going to label is the smooth endoplasmic reticulum. So the main function of the smooth endoplasmic reticulum is for lipid synthesis and detoxification of drugs. And in order for us to differentiate between the rough endoplasmic reticulum and the smooth endoplasmic reticulum is the presence of ribosomes. So ribosomes will attach itself to the rough endoplasmic reticulum, whereas in the smooth endoplasmic reticulum there's no ribosomes on it. So the next structure that we are going to draw is the mitochondria. So we'll use a different color for this one. We'll use this dark blue. We'll use this blue color. So mitochondria looks like this. And the main function of the mitochondria is actually a site for cellular respiration. Site of cellular respiration. And also generation of energy. Energy that is produced by the mitochondria is in the form of ATP, or known as adenosine triphosphate. So the mitochondria is like the powerhouse of the cell, and it helps the cell to run smoothly and provides energy to carry out active transport and all metabolic activities inside the cells. Next, we are going to draw the vacuole. And there are three main functions of the vacuole, but before we go into depth about the structure of the vacuole is actually a sac that contains cell sap, which is bound by a semi-permeable membrane known as the tonoplast. And the main function of the vacuole is that when the vacuole is filled of the cell sap, the vacuole will provide structural support for herbaceous plant, and it also stores food and waste substances such as glucose, amino acids, carbon dioxide. So the main function of the vacuole is to store food and waste substances such as glucose and amino acids, and waste substances such as carbon dioxide. And it also provides structural support for herbaceous plant. Now the next thing that we are going to draw is that we are going to draw the chloroplast. So we'll use a green color. So the chloroplast looks like this, and it has like shielding on top of it. So here is the chloroplast. The chloroplast contains chlorophyll, and it is also the site of photosynthesis, where photosynthesis happens. This is. Last but not least, we are going to draw the cytoplasm. So the cytoplasm is the site of biochemical reaction, where biochemical reaction happens. Now let's do a recap of everything. So on the outer layer, or the outermost part of the cell, we have the cell wall. So the cell wall provides mechanical support for the plant, maintains the shape of the cell, and prevent bursting due to high osmotic pressure. It is because the cell wall is made out of a tough substance known as cellulose, which is really tough, flexible, and really strong material. Okay, and then we have the plasma membrane over here. The plasma membrane regulates the movement of substance in and out of the cell. And then next we have the vacuole. The vacuole stores food and waste substances such as glucose, amino acids, carbon dioxide, and it also provides structural support for herbaceous plant, meaning that when the cell is turgid, it can provide support for the plant to stay upright. And the next thing we have here is the mitochondria over here. So the mitochondria is the site of cellular respiration, and energy is generated in the form of ATP through mitochondria. ATP is used and energy is used in the cell for metabolic activities and any activities that requires energy, like active transport, for example. The next substance or the next organelle that we have is the smooth endoplasmic reticulum. The main function of the smooth endoplasmic reticulum is to synthesize lipid and to detoxify drugs. And next we have the rough endoplasmic reticulum. The main function of the rough endoplasmic reticulum is to transport protein synthesized by the ribosomes. And the ribosomes produce protein, so basically it is where protein synthesis happens. And here we have the nucleus. The nucleus controls all activities of the cells, and it contains the genetic information. To be precise, the chromatin over here contains the genetic information, and the nuclear membrane, this nuclear membrane here, separates between the cell fluid and the nucleus fluid, where chromatins and nucleolus are located. In the middle we have the nucleolus. The nucleolus is a dense structure where RNA is produced, and RNA is really important to decipher genetic information from the chromatins and to attach itself to the ribosomes for protein synthesis. And next, talking about protein synthesis, we have the Golgi apparatus, where the process of modifying and packaging proteins from the ribosomes will be carried out at this organelle. And the new protein will be encapsulated inside a secretory vesicle, where it will go to the furthest point of the plasma membrane for exocytosis, which is a fancy term for meaning secretion out of the cell. And next we have the chloroplast. The chloroplast is the site of photosynthesis, basically because inside the chloroplast we have the chlorophyll. We will further learn about the process of photosynthesis in another video. Basically we are going to talk about the Calvin cycle. And next we have the cytoplasm. The cytoplasm is where biochemical reaction occurs. That's about it for today, guys. Thank you so much for watching this video. I hope that you can benefit from this video, and if you find this video helpful, do share it with your friends and follow me on my Facebook and Instagram for more tutorials and contents. That's all for today, guys. So see you soon in another video. Bye.